And we use an evolutionary framework. We compare the nucleotides. We don't compare percent similarities. We use a cladistic approach, character-based approach. And I'll get into that. So we actually preserve the nucleotides throughout the whole analysis, such that you can study biological data as it's presented. Okay? We're not converting to percent similarities, and those SNPs become mutations. Okay? And those mutations have function. So we're combining all that also in a geographic framework, putting evolutionary trees and key mutations and key other features, such as those I'm grouping in a set called field sciences, which mean the hosts of the animals, how the animals are husbanded, are they wild animals, are they domestic animals, and so forth. And really, it's up to the creativity of the investigators to use these tools. Computers don't do anything except what you tell them to do. Okay? And we've created an environment. Our contribution is a phylogenetic layer for GIS. Okay? So we take these tree-like diagrams, represents the evolutionary history of the isolates, the strains, and we project it, much like this projector is projecting an image on the screen. We project that tree and contort it based on the geography, and I'll show you that in detail. And that's just one layer that GIS professionals may use. Other professionals may use transit layers, where to evaluate a hypothesis as to, for example, are shipments of animals spreading disease? Do these lineages seem to spread along roads or rivers? Or do migratory birds spread disease? You can put a layer of bird migration patterns in Google Earth or ArcGIS or whatever geographic browser you want to use. And our goal is to contribute this phylogenetic layer, represented, let's say, by one of these users, but share that among many users, such that we build a collaboratory and to look at the multifaceted nature of emerging infectious diseases and their spread. So breaking up that Venn diagram a little bit into the component sets, you have here in blue the genomic data, the ACs, Gs, and Ts for each isolate. It could be bacteria, it could be viruses, it could be fungi, it could be different animals or plants if you have a natural science interest. And then we have geographic references. And these are very simple formats. I'll show you. They're just text files. This is a FASTA file. This is a CSV, comma separated value file. Comes out of Excel. And time is optional. It's very valuable, though. As you can see, I'll show you some animations. Okay? But you don't need time to calculate Supramap. And we have a web-based application. All you need to run it is a web browser and an internet connection. All the calculations are done in a computing cluster. We have one in Ohio. I moved recently from Ohio to North Carolina. It's primarily running on one in North Carolina now, I'm proud to say. And it runs an engine, really a compute engine is called POI, which is an acronym that doesn't mean a whole lot anymore, but it does an operation called direct optimization, where multiple sequence alignment and tree search are done confluently. And I'll break that black box open a little bit later for you. And Supramap is really a plug-in to POI, which calculates, based on the tree and the georeferences, what's called a keyhole markup file. Keyhole markup file, KML, is to Google Earth or other geographic browsers what HTML is to your web browser. Okay, it's the underlying code that says where to put the lines on the Earth, and those lines to us represent phylogenetic lineages. Okay? So the interface is really easy, and we'll go through this as well. You sign up; it's a free service supported by U.S. taxpayers. There's no charge to use it or anything like that. Typically in epidemiology, or from its origin, the view of the universe may be of the occurrence of disease or the occurrence of symptoms in patients. Okay? So you may see points on a map. Here, for example, this is an example. Let's say epidemiologists see influenza-like illnesses in Chicago. These are symptoms: headache, fever, maybe gastrointestinal, respiratory symptoms. And maybe we see similar symptoms in Toronto, okay? And maybe we see similar symptoms in Washington. And just because those symptoms are similar doesn't mean they're caused by the same underlying pathogen. And that's what we're getting at. We're flipping the coin of epidemiology around from syndromes in patients to pathogens in biology and evolution, okay? So, influenza illnesses. This could be influenza, just by looking at the symptoms. This could be SARS back in Toronto, and that distinction is very important. Okay? And this could be just a common cold in Washington. Or is it an outbreak spanning 2 countries? Well, let's say we did have the wherewithal to do some sequencing. And we got a little bit of pathogen RNA out of Chicago, out of Toronto, and out of Washington. So at the surface of the Earth, A, B, and C are actually observations of pathogens by virtue of their genome or marker genes that are interesting, MLST or whatever system you want. Okay? And we did some calculations. We searched for shared mutations. And now I want to store the fact that Toronto and Chicago are more closely related to each other than either is to Washington. Okay? I want to store that information in an inference, in a construct, D. Okay? So it's a mathematical midpoint for shared mutations. But what we're doing at Supramap, above and beyond phylogenetics, is giving this midpoint geography as well. So it's an evolutionary and geographical midpoint. And time here is represented, or inclusiveness, more by altitude. The altitude doesn't mean anything. I'm not using branch links in the traditional sense. I'm just putting the inferences as they include more descendants high up to distinguish inference from observation. Okay? And these are just arbitrary links. Right? They don't represent evolutionary distance or anything like that. Common ancestor D shares some mutations with the isolate out of Washington, and we store that in common ancestor E. Okay? So E is a midpoint representing the evolutionary set of Chicago, Toronto, plus Washington, and that occurs somewhere over Lake Ontario. What's really, really important at this point is to connect E to background data, to search GenBank, search your own archives in your countries if they're not shared publicly. You can do it through similarity search and then add data into ever more inclusive alignments and tree searches. And what you want to find out is, what does E connect to? Where are those pathogens coming from? Have they been sequenced before? What animals may carry them? And then what kind of biomedicine has been done on those pathogens? So you can use evolutionary inference to immediately open up countermeasures. Are there drugs that treat these pathogens? Which drugs are they susceptible to? Which drugs are they resistant to? Is it a benign infection or a deadly infection? And now that can be opened up by the search for the red line. In this case, we're going to study geography. Where did this pathogen originate? You can ask the question, does E connect to pathogens from Asia? Does E connect from pathogens from Europe or from Brazil, wherever you have data? Or did the pathogen originate in North America and spread abroad? I made this slide before H1N1, I swear. Does E not have any, is it a novel disease and not have any ancestors that were previously sequenced? And do we have to warn our public health partners in Asia, Europe, South America, and so forth that a novel pathogen is emerging in the Americas and likely to spread your way? A further concern is which hosts carry a pathogen. This will affect how pathogens are spread. And I'll show you examples comparing and contrasting human-borne pathogens versus animal-borne pathogens in our system. So there was a debate in early 2000s about which hosts carry H5N1, avian influenza. And those people who were concerned with bird conservation said, well, it must be agricultural practices that carried H5N1. And those people concerned with agriculture and had agricultural interests said, no, it's got to be the wild birds. Okay, so we thought we'd use our system to sort of get at this dichotomy and even examine if it really was a dichotomous kind of hypothesis. So we built a map. This is the same thing you saw before, but just more complex with real data. This is 300 genomes of H5N1 data. And here I'm adding another dimension, colors. Okay? These different dots are key to the colors. Most importantly, in blue, galliform hosted lineages of the virus, chickens, and red, anisoform ducks and geese hosted lineages. Okay? We don't have enough metadata at this time to evaluate wild or domestic geese. Okay? So some of these are husband, some are not. It's just we're using what's in GenBank. We've seen a lot of progress in that regard lately, but it's still a work in progress. So when you submit sequences, those sequences are not valuable unless you submit rich metadata with them. Here, the background data indicates, or we set it at a goose in Guangdong, China in 1996, and I'll animate this for you later. And so you can evaluate that putatively dichotomous hypothesis of wild or domestic birds by saying, well, I know chickens are domestic, okay? So it's not like anisiform birds where you have an ambiguity. And chickens don't fly. So clearly, you can see one line coming from central continental Asia to the Indonesian archipelago, and these are clearly chicken-hosted lineages. So in the context of Indonesia, clearly chickens or trading chickens is what's carrying H5N1 into the country. And that's an important thing to know, important distinction to make. You have to know how the virus is getting to your country before you develop some sort of policy to treat it. And that's why I disagree with the fact that Dr. Riley said that just mapping things doesn't teach you anything. That here's a direct, actionable piece of data from mapping things. Okay? Furthermore, Korea and Japan share chicken-hosted ancestry of their virus, and we're honest about what we don't know. Sometimes the algorithm cannot distinguish. Okay, the data is ambiguous, and sometimes we can resolve it with more data. Science isn't always a process, right? But be honest about what you have at hand. And so this yellow line represents ambiguity between China and the Korea and Japan cases. But locally, in Korea and Japan, the virus is being carried by chickens. Here, early on in the epidemic, in contrast to both of those, the virus is being carried in central China largely by ducks. If I turn the Earth a little bit, and I look across Eurasia, so I push China and Indonesia a little off to the side there, the right side. We're looking across Eurasia, India, the Arabian Peninsula, and Europe here. There was a case in 1994, Kundu like this one, where in customs in Brussels, they captured somebody carrying an eagle in hand luggage, and that eagle was infected with H5N1. That sequence was published, and we put it in our map, and it makes this large arcing green line. And that's really interesting because here you have this sort of virus over a decade hop skipping and jumping around Asia, making this huge leap from Bangkok to Brussels. Okay? This discordance between the huge geographic distance with the genetic similarity, this isolate's identical to the one on Brussels, we think is important to understand other processes besides migration or trade carrying viruses. Here's illegal trade. Here's smuggling, potentially moving a virus from Asia to Europe. Fortunately, it's customs, and the virus came into Europe through other processes later. Furthermore, we can show you, we can change the coloring, okay, representing other features. I was showing you host metadata by virtue of colors, and where we saw a color change, that was a zoonotic shift in the virus. Here I'm showing you mutation, just another kind of shift. In this case, from ancestor T to A, for example, or from ancestor C to descendant T. Okay, and a user in Denmark wants to know exactly what's happening in his location, and that's what he sees. These may be functional virus mutations, pardon me, and they may be just neutral mutations. Okay? Some people have been studying through genetic engineering and how H5N1 and now H7N9 can infect from mammal to mammal. These are avian viruses, but if you cause a certain set of mutations, you can get mammalian to mammalian transmission, which is a harbinger, if it occurs ever in nature, of a potentially pandemic virus. You can't see the legend here, but the wild type, the colors on here. Here, a user's browsing his particular region, a point. Mutations occurring at a point. Overall in the map in colors, in white, I'm representing in the PB2 gene, glutamic acid 627, the amino acid genotype. In red, lysine 627. when it's red, when it carries lysine, the virus can infect mammals. And this sort of spawned on us the idea that what we're doing is not necessarily, you can call it epidemiology or not call it epidemiology, depending on your biases, but what we're doing is sort of like meteorology, okay? This is a wave of viruses moving east to west in sort of a weather report of mammalian infectivity in H5N1. And a lot of people found that concept to be very useful. And I'm at the end of this set. Just go to home, proceed. Any